Alright everybody, welcome back. We're going to be doing Lab 7 today. Hope you've all been doing well. Today we're going to be doing gel electrophoresis and as well as root knot nematode extractions as well as observations. And then we're going to be looking at your McConkey plates from last week. So last week, if you recall, we did PCR, right? We amplified our DNA that we had and we got a whole bunch of copies. But now that we have those copies, or not, we can't really tell because if you look at your reaction tube, it still looks like it's just clear solution. You know, it could be water for all we know. So we need a method of separating the DNA out, and we do that by separation of size. We know where those primers are along the gene that we're targeting, and we know how large our fragment is, or is expected to be. So if we're able to separate those two out, we'll be able to tell, oh, this very large band up here, it's chromosomal DNA because it's all the DNA that an organism has. Whereas this DNA down here that's very small is the gene that we're interested in. And we'll show you guys how that works. We do that through a process called electrophoresis. So the reason it's called electrophoresis is because we use an electric current to separate DNA. What charge does DNA have on it? You guys recall? We know it's got a phosphate backbone. DNA has a phosphate backbone. Do you guys remember back in general chemistry or OChem? What charge do phosphate groups have? It's phosphorus and 4 oxygens. 3 or 4. Should be negative, right? Because it's got all those electrons on it. So DNA is negatively charged overall because of that. And we exploit that through electrophoresis. So we use something called an agarose gel, which it actually is quite like jelly. It's a gel-like substance. It's composed of agarose, which is this type of sugar that's refined from seaweed, and also a buffer. Here we're going to be using TAE buffer. The buffer is important because it has the ability to conduct a current, whereas something like deionized water does not. So the buffer has ions floating around in it that have a charge in them. Now the agarose, the way that this agarose is able to not split apart, but differentiate between DNA fragments of different length is because of its properties of when it's made. So you guys have ever made Jello before? You know it's very, jelly-like. Agarose gels are the same, except if you look at it at the molecular level, it's got pores in it, little holes in it. And these holes will impede the movement of DNA through the gel. So shorter fragments will travel faster through the gel than longer fragments. That changed automatically. The more agarose you use, so the higher concentration of agarose you put in a gel, the more pores it has. So it'll make the DNA a little bit difficult to move through it if it's longer strands. Whereas smaller strands will still be able to move through the agarose quickly. And I'll explain to you what I mean by that. So how many of you guys have ever watched The Price is Right? Or any of it? Well, you know what it is. There's this game that they have. It's actually my favorite. It's called Plinko. What happens is you got this board here. It's just a wall with pegs coming out of it, right? And a contestant walks up here, and he's got a little plastic chip, just a little disc, and he drops it. And as the peg goes through it, it starts hitting these pegs, and it goes plink, 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 and eventually it lands somewhere. That's why they call it Plinko. This board here can be considered a matrix in which you have pegs which form the holes through it. So now imagine you have this, right? Imagine you have a strand of rope that you have to pull through this matrix. If you have a long strand of rope, it's going to be tougher and tougher for it to pass through because it's going to get all tangled up in the pegs up here, right? But if you have a shorter piece of rope, would that be easier or harder to pull it through this matrix? It'd be easier, right? Because it's going to get tangled up less and less. And that's essentially what happens with agarose gels. So, I guess I should go back. with an agarose gel, you have DNA which serves as that rope, and the longer it is, the less quickly it's going to travel through because of the current. And the shorter it is, it's going to travel faster since it'll get less caught in those pores. Now, after we pass the DNA through the gel, it will separate it out by size. However, we still won't be able to visualize it, right? You won't be able to see it because it's all clear. The agarose, you'll see the gels in a minute when you load them up. They're very clear, just like what we're going to be loading into it. So we put a chemical in the gel called lithium bromide, which is a chemical that actually binds to DNA. And if you guys recall, DNA, if you unwind it, looks kind of like a stepladder, right? So it's got spaces between those bases. You've got your phosphate backbones with your bases in the center. In between those bases, if you have a chemical that's relatively flat and positively charged, because remember DNA is overall negatively charged, so the opposite charges are going to attract. If you have a chemical that's relatively flat and positively charged, it can squeeze in between those bases, and we call that intercalating. Lithium bromide intercalates inside of the bases. So here's what lithium bromide looks like. You can see it's very flat, right? Because of all these double bonds and all these rings. You really only have this ring over here that can rotate. But overall, when you put it in solution, this bromine ionizes, so it falls off. It takes the extra electrons from nitrogen here, and you get a positive charge on that nitrogen. That's what makes it overall positively charged. And you see it's very flat. You guys, who here's taking organic chemistry or something like that? Well, when you have a whole bunch of rings like this with double bonds, with all this resonance here, the overall shape of it is very flat. So you can think of all this as flat. And that flatness allows it to go in between the bases of DNA. So we call that intercalating. Now, the fluorophore. Fluo stands for like fluorescence or brightness. And if you've taken your organic chemistry, you remember with resonance, which is all these rings up here, you have a lot of resonance. The more resonance you have, the more energy levels that the electrons can move around about. You guys don't need to know that, but all that means is that lithium bromide actually fluoresces when you shine UV light on it. So why would we want it to use lithium bromide then if it shines underneath UV light? So you can see the DNA, right? So when we add lithium bromide to the agarose gel and then run the DNA through the gel, all the lithium that's sitting around in the gel is going to bind to the DNA. And so when we shine a UV light on it, we'll be able to see the lithium lighting up. But we'll see it lighting up because it's concentrated by that DNA. So what we're actually seeing with our agarose gels under UV light is not actually the DNA, but the lithium bromide. However, because we can see it concentrated in areas, we know, oh, this must be the DNA, because why else would all the lithium concentrate into local spots? It normally wouldn't happen. One thing to know though is that it can be carcinogenic. I don't know if it's been shown to, if it's been conclusively proved to be carcinogenic, meaning it can cause cancer. But if any chemical binds to DNA, it's probably not going to be very good for your cells. So, and I should have taken that out. I think we are using lithium bromide here. What that means is wear your gloves and don't touch the gel directly. So I'll show you guys how to load up the gel. So the way these gels are made, just for your information, you put agarose, which is like a powder, and you mix it with the buffer, mix it up, and you microwave it until it completely starts to boil, so all the agarose dissolves. And then you just pour it into a cast, a mold, and then it'll take the shape of the mold. So we put these indentations, which we call combs, so that it'll make wells, like a little cup for the DNA to be placed into. And then we just let it solidify, and once it's solid, we can use it for electrophoresis. So any questions on that then? That's the whole basic concept behind electrophoresis. Now I'm going to show you how the separation actually moves about, because it's a little bit complicated. Well, not really. hopefully it's not complicated when I explain it. Let's see. Electrophoresis, this tank, it's called the gel box or the tank. The gel's going to sit here in the middle inside this tray, and then we're going to fill up the tank with the buffer, in this case TAE. The buffer has to be completely, the gel has to be completely under the buffer in order for the current to pass through the gel. And let's see, samples are loaded via pipette into the well. These are the teeth down here, you can't really see it, the combs. And then we will always load a DNA ladder. I'll show you what a DNA ladder is in a second. Alright, here's a better picture. So you've got your gel, and you've got these wells, which are just indentations. We put our DNA into these wells, and then we run a current through it. Now, what is a current? Have you guys taken physics yet? A current is just the movement of electrons from negative, somewhere that has a lot of negative charge, to a positive charge. Why would we want to put the DNA closer to the negative electrode than to the positive electrode? Electrode just means something that moves the electron. So you've got electrons moving from up here to down here, right? Because electrons, what charge do they have? They have a negative charge, so they're going to be moving to this positive electrode up here. So why would we want the DNA to start up here and move down here? Why not start down here and move up?
They started once they go.
DNA, well, sort of. DNA, if you remember, has the phosphate backbone, so it's negatively charged. And opposites attract, but like charges repel. So if you have DNA with a negative charge, and you have an electrode up here, it'll start to move away from that negative spot because you got the electrons that are moving towards it and repelling it. So that's why the DNA will start to move towards the positive electrode. And then once we visualize it, you'll see that you have very discrete bands. Now remember, the slower something is, the larger a strand of DNA is when loaded into this gel, the slower it'll move. Right? So if we start up here and we move down here, you got two bands. You got one right here and one right there. This band down here, is it shorter or larger than this band up here? What would you guess? This one down here would be smaller because it moved through the gel faster than this band up here. Does that make sense? So that's how we differentiate between the different sizes, or between the different bands of DNA. We've also got something called a ladder. In this case, I don't know if it's a ladder, but a ladder is a whole bunch of different strands of DNA within one well that have a known sequence length. So we run this alongside our samples. Sorry, it's a little blurry, but we know, okay, this band up here, it's 1,500, 1,200, 1,000. And as you go further and further down the gel, you can see the length of them become smaller. And so we compare our own band to this ladder, and if we're expecting, say, like a 650 base pair band, we compare it to the ladder. This is kind of a bad picture. And we can see, oh yes, 700 is up here somewhere, and 600 is down here. So this band is right along the same length, or is about the same length that we would expect to see. And we can say, oh, this must be the gene that we're looking after, whatever DNA we amplified, because it's the correct size. So, does that make sense to everyone?
The way that gels are actually conducted, or
whoops, that is part of a different slide. This is just another schematic of what I just showed you. You've got your negative electrode, your negative node, and your positive node. And the way you remember which is which is you say run to red. The DNA is going to run to red. Right? And this applies with any type of electricity related stuff that you do. Your negative cable is always going to be attached to the negative, your black cable is going to be attached to your negative node. Your positive is going to be red. So if any of you ever worked on cars and you look at the car battery, you'll see a black cable and a red cable. And you'll know which is which. Or, excuse me, a black electrode and a red electrode. See, larger DNA molecules have a harder time passing through the pores. Smaller travels faster because it travels through the pores. Okay, so this person is running the PCR and expecting a band size of 650 base pairs. So they've got these 4 samples down here. They've got their positive control. You see a band there. You've got their negative control. Negative means they don't expect to see anything. And you don't see anything. So what would you conclude from this experiment here? Was this PCR successful or unsuccessful, and why? Anyone? All right, I'm going to have to pick names. Let's see, first one I see. Jillian. Would this be a successful or unsuccessful experiment? And why?
It would be successful because all of the samples match the positive control and the negative control has nothing. So then you know that, I guess what you're seeing is from the samples. I guess,
Yeah, exactly.
And it matches along with DNA around it as well.
Right. So for those of you at home that probably weren't able to hear, she said that because the samples are the same size as the positive control and your negative control came out negative, it would indicate that this was successful. And then here, this is some of my work. This is what I did last week. Just to show if you remember with PCR, the amount of DNA that you start off with will determine how much of the amplicons you have. Another name for these bands are the amplicons because they're amplified DNA. And I was trying to see what the detection limit was by putting in various amounts of picograms of cDNA. So you got 10 picograms, 1 picogram, 0.1 picogram. My negative control is this well here, and my positive control is this here. So would you say I was able to amplify the correct band based off of these results? I would, right? Because the positive control is there. It's the same size as, my samples are the same size as the positive control, and my negative is completely missing. So that would be something successful. One of the very few successful experiments in the lab. Okay, so any questions then on electrophoresis? Now we're going to move on to the nematode portion. If you guys remember lab 4, so 3 weeks ago you guys inoculated some tomato plants with nematode eggs. Now by this time the nematodes have hatched and hopefully started to infect the roots of the tomato plants. Now we had 2 varieties, one was resistant and one was susceptible, or that's what we claimed, but today we're going to see if that's actually true. So what you guys are going to do is we're going to take out the roots and try to extract the female nematodes that are living inside the roots. So here's the protocol. I'll show you guys how we do everything right now, but just to quickly summarize it, we're going to take the roots out of the plastic tube that you have. Try not to break the roots off. We're going to clear the soil off into those trays that we have here. You can rinse off the roots with water, and then you're going to write down your observations. So don't forget to write that down as part of your assignment. Take a look at the lesions, not lesions, galls, or the size of them. If you see anything weird going on with the foliage, any signs or symptoms. Then we're going to cut off the roots. We're going to dump them in bleach, 10% bleach, for about 10 minutes. And afterwards, we're going to dye them with some red food coloring. We're going to microwave the roots with the red food coloring so that the food coloring acts as a sort of dye so we can differentiate between the root cells and the nematodes. And then afterwards, we're going to look at those roots underneath the dissecting microscope and see if we can't find any of those female nematodes. So that's the experiment in a nutshell. We're also going to be loading your PCR reactions into an agarose gel so that we can see what's what. And we're also going to look at your McConkey agar from last week and take observations on that. So any questions then before we start the technical part? No? Okay. Let's start up.